Good morning, everyone. My name's Nick Mancuso. I'm super excited to be here today to chat about variational inference for large-scale genomic data. So this talk is like kind of in a new direction for me. It's still related to a lot of what I've been thinking about and doing, but we'll see how this goes, I guess. Right. So just like a show of hands, like who's familiar with the concept of variational inference from the get-go? So like a good bit of people. All right, excellent. So if not, like no biggie. We're going to try our best to wade our way through these things and then hopefully look at some results to place these things in context. But like, I should be somewhat upfront in that, the title is Variational Inference for Large-Scale Genomic Data, but, I'm going to make a last-minute change and put parentheses around that, and because there will be some aspects that aren't necessarily variational, but they're all tied in for the most part. I know this is probably like the millionth time we've seen something along these lines of this talk, but just for completeness sake, genome-wide association studies are arguably one of the most popular ways to assess whether or not genetics contribute or associate with some complex trait or disease. The way they work is by collecting either a large cohort or assembling some biobank, quantifying the genetic variation through some genotyping assay, or maybe more recently with low coverage whole genome sequencing. And then what we do is just kind of march along the genome, right, and test for association between genetic variants and the disease outcomes. And so, we've at this point, we're like, what, 10, 15 years into this. So we've amassed like a humongous catalog of associations across all sorts of traits, diseases, biomarkers, like drug use, like you name it, like just like, throw a dart at a wall and that's probably some phenotype if you have like phenotypes on a wall for some reason, and there's going to be a GWAS for it. And so, what it provides essentially from GWAS is unfortunately for geneticists and disease researchers are not causal mechanisms. Overwhelmingly, what we find are regions associated with the trait or focal disease. And so what we're left with is kind of trying to figure out what the mechanisms driving disease risk are. Along the way, this is not surprising right now, but along the way, most of the associations are non-coding. Again, it's one of those things, throw a dart at the genome, it's going to fall into non-coding, overwhelmingly so compared to coding. But still, given that, there's an abundance of evidence showing that these associations are in non-coding regions that are enriched for functional activity, and so it suggests that there's some regulatory principles at play that mediate risk in some manner. And so just what we're showing here is a little snippet from a schizophrenia GWAS from a few years back. And so again, each point corresponds to some association strength of a SNP, and everything below it is the kind of like all the genes that we know of at that locus. And so if we wanted to eyeball it, like we really would it would be hopeless to try to identify what the causal gene is. And so, we're really reliant upon additional evidence in some way, shape, or form to kind of figure out what's happening here. And so, we've seen already from an excellent tutorial last week where folks have demonstrated how to integrate molecular and functional information together with GWAS to identify and understand disease mechanisms. And the primary approach works in that you have some reference panel where you have functional information, be it gene expression or some other like epigenetic marker. We fit some penalized linear model to that feature and then predict it into GWAS cohorts and see if it tracks with disease risk. There are certainly plenty of caveats that come with that, but provided that we see an association, right, that's like a great sort of next step to follow up with additional approaches. Right, and so we've already kind of understood by this point now that GWAS links genetics with disease risk, and so like the one way to understand disease risk is through that like bottom-up approach, right? We have these associations, and we'd like to sort of integrate molecular data or functional data to say like, well, what are the mechanisms? What are the genes? What's happening here? Sort of an alternative way to possibly understand what's happening with genetic risk regions is to take maybe like a top-down or like maybe a horizontal, I don't know, I'm losing kind of the metaphor in terms of direction here, but integrating high-level phenotypes to get a sense like maybe what the etiology is, right? And so here, probably the issue is that like maybe too often, right, genetics associates with the phenotypes or disease risk, and so this is like the latest GWAS catalog, and so it's like completely uninterpretable, right? Just like, again, any genetic variant is going to be associated with something somewhere. And so, what do I mean by that maybe a bit more concretely? We can integrate phenome information to understand disease mechanisms, right? So there's been a couple of studies that go ahead and just take all of our association data and try to distill it from like a top-down approach to understand what might be going on, right? And so, like, hopefully by now, what I've demonstrated to you all is that there's sort of like two broad approaches to maybe understand GWAS risk regions from like a scientific standpoint, right? But then there's also kind of just like a practical issue of scale. And so global biobanks sort of really reflect the massive scale of available genome and phenome, right? And so here what I'm showing is like a figure from a recent preprint from the Global Biobank Meta-Analysis Initiative. And at this point, it's really reflecting the contributions from more than 2 million individuals. And so, there's not going to be a situation where you have maybe 2 million individuals in front of us on a cluster that we need to analyze, but it's more or less really meant to capture sort of the computational challenge that's in front of us, and like how do we analyze large-scale data, right, with hundreds of thousands, millions of individuals, thousands of phenotypes, or tens of thousands of molecular phenotypes. And so these large-scale genomic analyses really require scalable inference. It's something that just we can't ignore, and the field hasn't really ignored it since day one. That's kind of been the theme from day one. And GWAS researchers, like among many others, have continually met this challenge, right? And then pushed the field forward in terms of how do we infer the parameters of interest, right? How do we even just come up with a point estimate or some estimate of the parameters we're interested in when we have this much data to analyze, right? And so, the field has really pushed the state of the art in mixed models, covariance and variance components, variable selection, with just a few examples like with Bolt LMM, Sage, REML, SCORE, and some other methods. And then similarly, right, the field has done an excellent job about developing methods that leverage summary statistics that also enable kind of fast, scalable inference for population parameters. And so, but this list is by no means exhaustive, and so my apologies to all the hardworking folks that are developing methods in this area. But I think just that's really just one fantastic aspect of our field is that there's just been a lot of thought put into how do we make these things scale. And so, with that in mind, again, the topic of this talk is variational inference, and so, I've kind of hopefully set the stage. And so, the next thing I want to talk about is have you heard the good word of our friend, Thomas Bayes? And so, I actually, learned more recently that we don't even know if this is what he looks like. This is just some illustration that was in some early journal or book. And so this is our best guess, right? Maybe he looked like that, maybe not. And at the end of the day with inference, right, and in a Bayesian setting is that given some data, inference in general, given some data, we'd like to infer some parameter of interest, right? And this well-established and known Bayes theorem says, how can we sort of quantify the uncertainty in our parameter upon seeing the data that we have at hand, right? And so here what we have is just the posterior for our parameter of interest. We have the likelihood, some prior over the parameter, and then the marginal at the bottom. But really, probably this form, what we know now, isn't actually maybe what we would attribute to Bayes. It's probably more the result of Pierre-Simon Laplace. But you know, fine. We'll just keep running with it. There's like a law by now that, you know, like theorems and names of things aren't named after the person who came up with it typically. And so what's the problem, right? So this is like why this? This is like, you know, why do we need Bayesian inference? Well, you know, I think it provides like a really natural framework in terms of either integrating or like providing structure to our parameters, right? Oftentimes we know a lot about sort of the parameter space, and we don't necessarily need to just assume that we know nothing, right? We can constrain ourselves to either place effects closer to 0, or maybe we have some functional information that we would believe like we should push effect sizes larger in some parts of the genome. And so why not just do this all over the place, right? Like, why not just like let's go crazy with it? You know, let's develop Bayesian everything. But the problem is, like, you know, it's very rarely is it the case that this sort of structural form is computable in closed form. It's like only in limiting cases. And so oftentimes what happens is that integral on the bottom is like exponential in time and data. And so it's just like we have to throw our hands up, right? And so what can we do? Well, so that's where variational inference is sort of a modern approach to kind of tackle that, right? This isn't the only such approach to get approximate inference in a Bayesian setting, but it's my talk, so I get to talk about what I want, and this is what we'll talk about. And so, all right, so what is variational inference, right? It's an approach to do approximate Bayesian inference, right? Or how I learned to stop worrying and love just approximate posteriors. And so, you know, can we approximate, the goal here is like, can we approximate this posterior with some surrogate distribution, right? If we've established that like directly computing our posterior is challenging, we can't do it. Let's say like, okay, fine, can we come up with some other function q that's like close enough, right? That like we can use in practice and still learn something of interest that we can provide to our, you know, wet lab colleagues to like follow up with or something, all right? And so how do we do that? Well, you know, let's come up with an objective and say we want to find some function q star that's the result of like this minimization scheme, right? We want to find some q that's close to the true posterior, where close is measured in terms of the KL divergence. And so the KL divergence is really just some kind of fancy form just to say like how dissimilar is one statistical distribution from another. And the way we can write it out is just like the difference in like kind of the log probability minus like the log true posterior, where the expectation is with respect to like our approximating distribution, right? And so we can write this out. Unfortunately, though, those with keen eyes might note that like this objective is still hard. We haven't really done anything. We've kind of set up this framework, and we were left like off in the exact same spot that we started, and that the goal is to find some q that's close to the true posterior. We can't even evaluate this because the true posterior is intractable in the first place, right? But fortunately for us, that the marginal likelihood, or the hard part of this, is constant, and so we can actually compute something up to a constant. But just real quick, just for a note, like as an example, like what the heck is this KL divergence, right? Here we have like two Gaussian distributions that have different means and maybe the same variance. And down here we have two different Gaussian distributions that have the same means as the above examples, but wider variances. And so intuitively we would think like this bottom pair of distributions are maybe closer than the one up top. And so that gives like kind of maybe, depending on, you know, how you want, but at least in terms of KL divergence, it's close enough. And so this gives us like a sense of like closeness, right? Where as if we just took the parameters themselves in terms of the means, right, they wouldn't be any different. Okay, so great. And so what we can end up doing is just rewriting the objective using this thing called the elbow, or the evidence lower bound, where essentially we're just looking at the log joint of our data, right, which is the likelihood in the prior, minus this approximating distribution. And what we'd like to do is maximize this lower bound. And what's nice about this is actually you can reformulate this as like a penalized expected maximal likelihood. And I feel like that also provides like a very natural interpretation of what's going on here. Is if we just do some symbol pushing and move things around, what we get is the expected log likelihood and the KL divergence with our approximate or surrogate posterior and the prior, right? So you can think of as it, we want to find parameters that fit the data well through the likelihood, but we don't want to go too far from our priors, right? And so that's what's penalizing it. Are there any questions at this point? Man, this is, I've spent a lot of time. I need to go faster. Okay, so shouldn't this just result in our true posterior, right? If the goal is to like find this Q that gets close, if it's at 0 or like at some constant, wouldn't that just provide the same, like a shortcut to the hard problem of just like approximating the posterior? Well, the problem that is, well, not the problem, but what I didn't go into is that there is like a constraint on the distributions we want to look at. And so here in orange, we want to find some Q that's in some like family of distributions Q. And like what we typically do, or what researchers typically do in practice, is just have some factorization where we say, okay, if we had some K variables we wanted to learn, now they're just all independent in our model, right? And so what that does is affords us sort of the computational tractability, but also it comes at a cost. And so does anyone want to take a guess at what that cost may be in terms of inference? Well, I'll just jump to, since we're running out of time. But the goal is by constraining things to be independent, right, we lose any sort of benefit of correlation in our parameters, right? So oftentimes in the posterior, things will be correlated. And so if we're constraining our approximate form to be independent, that sort of information across the parameters will be lost, right? And so that may or may not affect inference for the problem at hand, right? If our goal is just to infer some mean parameters, it might not matter. But if our goal is to infer things like variance components or something in a higher moment space, then it might be an issue. Okay, so by now, like what I've established or hopefully gotten the point is like, all right, what we want to do is find some fast inference scheme. We want to go a Bayesian route because of the additional structure we can sort of imbue into our problem. And we want to do it quickly using variational inference. And so I've sort of set up this framework, but like how do we even go about finding these things, right? We have this nice objective, like what do we do? And so one of the simplest approaches is the reason why it's called variational inference in the first place is that using some nice math from variational calculus, we can find like the optimal form for each Q is just going to be proportional to essentially like integrating out everything else, right? So if our parameter of interest is some theta I, we just keep everything else fixed, integrate it out under expectation, and it'll be proportional to that. Usually that will result in, after a lot of math, like some nice form that fits with our model. That won't always be the case, but in many contexts it will be, or at least for this talk. Let's see, so I need to, oh, I still got plenty of time. All right, so one nice example is so in TWAS, sort of the primary means to track associations between molecular or functional data with GWAS risk is to first fit a predictive model using some sort of cisgenetic locus around the functional feature. And so, some of the early papers established really nicely that using these Bayesian sparse linear mixed models do an excellent job of sort of handling the sparsity at a locus and predicting sort of the cisgenetic components of signal. But the one downside with that approach is that it's computationally challenging. It's reliant upon sampling schemes. And so, in theory, if we want to compute the exact posterior, it'd be exponential with respect to the number of SNPs. The sampling scheme won't be exponential, but it could potentially take a long time to sort of mix and get sort of accurate enough expectations, right? And so here the idea is that we just have some linear model, like gene expression. We want to infer these betas that are going to be sparse. And so a few years ago, there was this excellent paper that came out proposing a model called SUSY that's the sum of single effects. And so it took a different approach to thinking about these sparse Bayesian models. So rather than just saying there's this point slab on each SNP and we need to sort of figure out this giant mixture, we just assume that there are L effects. And the goal is to say, well, for each effect, which SNP is driving that. So here again, we have the same linear model, but rather than just a point slab, we say that beta is just a sum of some L effects for individual SNPs. And so here, this can factorize into just over each of the L terms, and where those are sort of going to be further factorized into a joint for the variables that select the SNPs, as well as like the effect condition on what variable is selected. And so after some symbol pushing in math, what like pops out of this is like a nice closed form for the approximate posterior for the effect, and same thing for like the posterior expectations. And in this scenario, computing beta will be linear with respect to the number of SNPs. Okay, so that's like a bunch of math, like, and not many like results in hand, right? And so I think like theorems and math are all well and have their place, but like we want some results to like sort of convince ourselves this is a reasonable thing to do. And so we did that a while back when GTEx V8 came out to trainees in my group. Wanted to compare the predictive performance of using the SUSY approach versus BSLMM and GTEx V8. And so what we did is we just randomly sampled genes through some tissue and locus, and GTEx focused on European ancestry individuals, and then compared the out-of-sample cross-validation R-squared, right? So pretty straightforward. And what we found is that actually like SUSY does really, really well compared to BSLMM. Right? So here what we're showing on the x-axis is like SUSY R-squareds, and on the y-axis is BSLMM. And we see like essentially like just a linear fit, right? Like they're doing like the same job, right? It's going to be hard maybe, you know, if we really drill down to say one's better than the other. But where it stands out is that, you know, if we want to fit predictive models for all of GTEx, there's like 1.2 million genes in total across all tissues. And so using the HPC, that, you know, would have taken like a couple of months. Right? But with SUSY, like we can do all of that in a week. And so that's great, right? Now we have this like super fast method to do predictive modeling that like we can fit in a week. And so all of our colleagues are happy because we're not like using the entire cluster for some time. So when does this talk goes to 11 or 11:15? Okay, good. So we got plenty of time. All right. So we leverage these predictive models in our approach, like called like multi-ancestry focus. The idea is here we'd like to do TWAS and fine mapping across like multiple ancestry groups. And so again, the idea is that we have GWAS from multiple ancestries for a given phenotype. We have the EQTL prediction weights, in this case fitted with SUSY, LD references, and then TWAS results, and we can compute this correlation and spit out for each gene like what the posterior probability that a gene is causal across both populations. And to give like an intuitive understanding, so here rather than LD, we have this sort of predicted expression correlation. You know, maybe in African individuals, it won't be as tight compared to the European ancestry counterparts because of not as strong LD patterns. And so what we see here in red, just for this toy example, is maybe the causal gene. Some other genes show up as significant just because of tagging in either population. But in sort of a joint model, what we can do is fine map, assuming that the gene itself is just shared across populations, which is fairly reasonable. And we can improve the performance to sort of place a higher posterior probability on the gene that's causal. And so the way this works is like we can model the sampling distribution across multiple ancestries just independently, given some causal configuration C, where that's just similar to, you know, those who are familiar with caviar. It's a very similar sort of structure, but here we're operating over like predicted expression rather than and genes rather than SNPs. So I'm going to skip ahead a little bit just for time reasons. But we can compute similarly the pips for each gene and credible sets over genes rather than genetic variants. We applied this to large-scale application in blood GWAS from Chen et al. So there was 15 blood phenotypes that were analyzed in multi-ancestry data a couple years back. However, there was like a large sample size imbalance. And we'll see how that plays out for results in a bit. And to do the TWAS, we fit prediction weights in Genoa, which again was a multi-ancestry cohort consisting of African Americans and European ancestry individuals. We used 1,000 genomes LD, and what we did was perform TWAS. compared against single ancestry focus as well as like some simple baselines. Looking at the TWAS results, we identified approximately 6,000 genes, 6,100 genes for EA, or European ancestry and African ancestry individuals. And this is like, again, largely the result of like differential power, right? We have like, 40 times sample size difference from one group compared to the other. And so I don't want to give the impression here that, genes matter for one group and the other, because that's certainly not the case. This is largely a function just of sample size. And so what we did was focus on the regions where we have comparable power by focusing on where there's like joint association signal to perform fine mapping, right? And so this may sort of tilt the scale in our favor by looking at regions where we know genes are likely to be shared, but for now that's what we focused on. And so one of the things we found is that TWAS effects correlate 20% more strongly across ancestries than GWAS. I think this result's largely consistent with other gene-based methods, where the idea here is you can think of just after properly accounting for LD or MAF differences across groups, we see more consistent effect sizes across the ancestries than just sort of the naive like SNP-based approach that doesn't account for like LD and minor allele frequency. So the next thing to do is we wanted to perform some amount of validation, which is challenging in this scenario. And so what we looked at, given that we're focusing on blood phenotypes, was say, well, given the credible sets computed by our approach here in red, the single ancestry approaches in yellow and blue, as well as like a naive baseline that just meta-analyzes the GWAS and uses the European results, we find that MA focus results are more enriched for hematopoietic categories compared to any of the other traits. And then similarly, we compared with like some recent approaches that the silver standard analysis, where we took the posterior probabilities assigned by each approach to see which better predicted monogenic or oligogenetic blood traits from OMIM data. And then we saw that like across all data sets that MA focus had the highest area under the receiver operating curve. I'm going to skip ahead. Okay, so that's kind of like the bottom-up approach of using predictive models and functional data to identify sort of disease mechanisms. But like coming at it from the flip side of like the top-down approach, there was this really nice paper that proposed this method a couple years back called GWAS, where you can take just a matrix of association statistics across many phenotypes and perform SVD to get a sense of what sort of latent or pleiotropic structure is present across like the phenotypes in hand. And so if we have this matrix Z, we can think of performing some SVD analysis or truncated SVD analysis where we look at like maybe the first K principal components or K singular values to decompose this matrix, where K is much, much smaller than the number of phenotypes. With this model, though, what we're left with is like, what are the actual model parameters and how should we interpret them, right? And so one approach that we came up with was a factor analysis model from effect sizes. And so here you can model the inferred effect sizes from GWAS as sort of a factor model in like a latent pleiotropic space. And so what we can do is go ahead and say that there's some, for any given phenotype, there's some latent set of directions, as well as like individual SNP loadings for each SNP and each like pleiotropic factor. And we can place a bunch of priors on these to constrain the structure. And then, if our goal is to do inference here, the true posterior is intractable. However, we can infer a surrogate just similar to the mean field model, where we go ahead and sort of kind of break all of these model parameters up independently. And so we took this method and analyzed more than 2,000 phenotypes and 50,000 variants from the UK, pan-UK biobank data, where we restricted SNPs to have at least one genome-wide significant SNP. And this resulted in, we can think of roughly like a 2K by 50K matrix now that we feed into both GWAS and our approach. And so what we found actually is that for Comparing across the two approaches, that our approach FactorGO leverages information to a greater extent from underpowered studies compared to the truncated SVD. And so what we're showing here is like this notion of a contribution score where each phenotype has a score about how much it's represented in the data, and we can compute the ratio of that score from our approach versus TSVD. And here, the color indicates sort of the sample size of the original GWAS at hand. And so when we see that sample size is large, they roughly are finding sort of the same information. However, when we get to smaller data sets, we see that sort of the information or representation of the phenotypes is much greater in our data set. And we believe this is like due to the fact that we're directly modeling effect sizes along with standard errors rather than some sort of like collapsed magnitude of association, right, where that sort of nuance may be lost. And so, for example, here we're looking at sort of the first factor from each method, and we see that the pleiotropic dimension from FactorGO captures the relationship of COVID-19 and BMI, where we see COVID-19 positive in two different GWAS in the UK Biobank data, followed by like numerous BMI or fat mass related phenotypes. And so this sort of reflects like with what we've seen from like broader scale analysis of just like genome-wide genetic correlation, where these things correlate at the genetic level. Whereas like the TSVD factor 1 completely misses this relationship between COVID-19 and these others and focuses mostly on the morphological traits that are related to like sort of fat mass and BMI. And again, I think this is primarily due to that effective sample size, right? That like for COVID-19 and Pan-UK Biobank, the effective sample size is very low because you have a few number of cases, and so our approach is able to tease that out to a greater extent. And so like maybe it's like easy to like wave our hands and say like, look, it's finding these things, it's doing a better job because we found COVID or something. And so we need to do some form of functional validation, and what we did was take 707 functional data sets that capture transcription factor specific and cell type specific like binding properties from the impact paper. And what we did then was focus right now, just one example for height. And so we looked at what the most important factor for height is in each data set. And so here from FactorGO, we see standing height at the top, followed by a bunch of mean carotid measurements. And similar for like TSVD, right? We see like standing and sitting height with some other like forced vital capacity, like maybe lung measurements. And then what we did is like take the loadings inferred from each approach and under some transformations see like do we see better enrichment of like these loadings, which we can think of as like kind of approximately as SNP effects with respect to these functional annotations. And like what we found is that FactorGO finds like a much greater enrichment at functionally relevant genomic annotations for height. For example, if we look at like, it might be hard to see here, but like, vascular related annotations, heart, bone, and muscle. Sorry, it's like a little bit harder to see, but maybe the second point's sort of eke out past the enrichments identified by the truncated SVD. So that's all well and good that we found sort of those results for like a quantitative trait, but next we wanted to see if like, does this hold for disease traits where there may be like more of an imbalance in results. And so we focus on like rheumatoid arthritis. And so here FactorGO pulled out several biomarkers, albumin, calcium, but however it also pulled out a bunch of like osteoporosis related phenotypes. Which seem to track with like observational and epidemiological studies that osteoporosis is a comorbidity for rheumatoid arthritis, and the leading factor for truncated SVD is like largely contributed by like IGF-1, which makes sense still in its own right? That's like a great biomarker for inflammation, and so we're not necessarily saying that like these should be capturing the exact same thing. They might be sort of independent components in different directions. And then when we look at functional annotations in this space, again, we see that FactorGO identifies greater enrichment for like relevant genomic annotations, such as blood. Yeah.
On the previous slide, like on the right there, you say TSVD factor 59.
Yeah.
So the IGF-1, that's the leading loading on the 59th SVD. You know, so what?
Yes.
How did you select the 59th one to show? Or?
Yeah, right. So what we did was for rheumatoid arthritis, we computed its contribution score to all of the factors and looked at which factor had the greatest for each method, right? And so here in FactorGO, that would have been factor 86, and for TSVD, that would have been factor 59, right? And so, in the relative scale, it seems like maybe this was placed higher up within TSVD, comparatively, but within that factor, then we see what traits are represented by the contribution scores, right? Right. So you can think of this bipartite graph, we focus on rheumatoid arthritis, what's the greatest factor? Given that, go back. What are all the factors or traits for that factor? Okay, and so there was a lot of work that went into this in terms of optimization, and so we leveraged GWAS, which comes with some really sort of powerful niceties to speed things up. And if we just did a very naive implementation in Python, this would take several hours to run in our Bayesian setting. However, with some additional techniques such as just-in-time compilation and flipping a single switch to get it to run on a GPU, we had effectively the same runtime as just doing a truncated SVD, which is, as anyone who's done that in practice knows, it's blindingly fast. It's really easy to do that, and so. What we have here is a sort of model that spits out parameters that are relevant to effect sizes, at sort of pleiotropic sense, and does so at a speed that's comparable to just truncated SVD. So this is kind of work in progress. So we're hoping to get this out soon. I'd love feedback from anyone, and happy to chat. So I think we'll probably stop there. Large-scale analysis of genomic data requires scalable inference. Right? I don't need to say that. Everyone who walked in this room knows that, right? It's nothing new, but it's kind of going to be always the case, right? There's always going to be a need for faster methods that as this data sets grow, and by now hopefully I've somewhat demonstrated that variational inference is a scalable, flexible approach to do approximate Bayesian inference. It's certainly not the only means to do fast inference. There are a lot of approaches, and sort of as demonstrated by the community in either a non-Bayesian setting or a Bayesian setting, and focus on applications in EQTL prediction and these pleiotropic factor analysis. Hopefully, we've demonstrated that things are fast and give reasonable enough results that we can use these things. So there's a huge thanks to everyone in my lab, especially those that worked on this, as well as collaborators and our funders, and those who are also at USC. I'm happy to take any questions. Okay, no questions. All right, so with that note, I will say I just want to take this moment to advertise that the Center for Genetic Epi at USC is hiring at several levels. So both at postdoc levels for various folks there, but also at assistant and associate professor level. It's come to sunny LA. There's a lot of exciting science here, and tacos are abundant and the beach is close. So. Hopefully those jointly can convince you. So cool. Thanks everyone.